Welcome to the Chemistry, Biology and Math Revision Hub. Today we are doing the Pearson Edexcel International A level Biology Unit 4 for June 2020. I have already done the part 1 of this paper, so this is the second part. And I'll put the link to the part 1 video below the description box of this video. Let's begin. Question 5 says, viruses can infect bacteria. Which virus can infect bacteria? So here they want to know which among these can infect bacteria. As you know, Ebola virus, no. The HIV virus does not, and the tobacco mosaic virus doesn't. So it's only the lambda-phage that can infect bacteria. So the answer to this should be a C. The next part says, some viruses that infect bacteria cause the production of molecules called holins. Holins form protein channels in the cell membrane of bacteria, and this allows polar molecules called lysins to reach the cell wall by facilitated diffusion. The DNA of these viruses codes for lysins. Describe the role of channel proteins in the facilitated diffusion. So here we know, since we are talking about facilitated diffusion, so we have to talk about movement from higher to lower, or following the concentration gradient, or you could say down the concentration gradient. And then we have to talk about how these proteins are going to be arranged, the ones that create the channel. So the channel proteins, how they're going to be arranged within the cell membrane. So I said the channel proteins provide the polar channel within the hydrophobic cell membrane. The polar channel facilitates the passage of the polar lysins down the concentration gradient. Remember, if this is the cell, let me try to do it like that. If this is the cell membrane, some of these channel proteins are going to be aligned within, but since we have the hydrophobic tails, the hydrophobic parts of the protein are going to be aligned facing the hydrophobic parts of the phospholipids, and that allows for the hydrophilic parts to be inside or the polar parts to be inside, creating a 3D structure that allows substances to go through. But the inclined molecules that are inside here are going to be hydrophilic, or you would say they're going to be polar, so that the polar molecules can go through. So the inner parts, the parts that we have in here, are going to be polar, or I would say hydrophilic, to allow the polar molecules to go through. But the outer parts, let me use a different color here. The outer parts on these proteins are going to be hydrophobic, so that they can align themselves with the hydrophobic tails of the phospholipid bilayer. So let's continue to the next part. Here they say, explain how the primary structure and the tertiary structure of holins, remember these are proteins, determine the properties of these channel proteins. So everybody knows the primary structure is a sequence of amino acids. They can be arranged in any order depending on the information in the DNA. So I say this arrangement determines the tertiary structure. Remember the primary structure having a sequence of amino acids with different R groups along the polypeptide that has been formed. This is going to fold into the secondary structure, and that folding creates for either hydrogen bonds or disulfide bonds. And when these bonds are being formed, the folding further folds into a tertiary structure. And the way the tertiary structure is going to appear will have depended on which R groups were closer to each other during the folding, and that would lead to the formation of the channel protein. So I say, since this determines the position of certain bonds like hydrogen bonds or disulfide bond, etc., within the structure, the polar R groups are arranged facing the channel, and the non-polar R groups are arranged facing away from the channel towards the hydrophobic tail of the phospholipid. So that could be okay for this part. Next they say, lysins break down the cell wall. Remember, this is a cell wall and not a cell membrane. So if these lysins break down the cell wall of bacteria, they say explain the role of lysins in the lytic cycle. You guys know in the lytic cycle, this is when these viral particles are going to be getting out of the bacteria that they had infected, or the infected bacteria. It means the cell wall has to give way so that these virus particles can go through. And to do that, it means the lysins are going to be breaking some bonds, separating the molecules within the cell wall to allow the pathway of these viral particles. So I said, during the lytic cycle, lysins break the bonds between the peptidoglycan molecules within the cell wall, and this allows the virus particles to get out of the bacterial cell wall once they have been formed. This brings us to the end of question 5. Let's continue on to question 6. Question 6. The photograph shows a ground squirrel. So this is the squirrel we are talking about. Let me continue down here. They say, ground squirrels feed on a variety of foods, including nuts. Ground squirrels have evolved food pouches. So they show you the food pouch, which is that. They say the photograph shows the food pouches of a ground squirrel. So let's go to the next page. So they say the table shows some information about 3 types of nuts. So they're acorns, hazelnuts, and walnuts. We can look at the table to see some information here. They've given us the acorn, and you can see how it looks like. They've given us a description of the length from the least possible to the highest, as well as the width and the energy content. The hazelnut looks like this one. We have the length, we have the width, and then they've given us extra information that this is a hard covering and the energy content per 100 grams. Next, we go to the walnut. We can see it looks like that. They've given us the dimensions, length as well as width, and they said this is a hard shell, and this is 2822 kilojoules per 100 grams. So we go to the next part. Again, that is information that we're going to need in the next part of this question. So they say the volume of the smallest walnut is approximately 50 centimeters cubed. Calculate the approximate volume of the largest hazelnut using the formula here. So they've given us that volume is equal to 4/3 pi L W squared. They've given us what pi is. They've given us the length, and then they've given us everything. So I'm just going to fit into this equation, keeping in mind that they want the largest hazelnut. So you go back to the hazelnut, and since we are inputting length and width, we have to go to the largest. Down here, you can see that is 2.5 as well as 1.5. Those are the values we're going to use. Putting them into this equation is going to be 4/3 times the pi, which is 3, times the length, which is 2.5. Just a minute, sorry. Let me take it back. Okay, and then this side times, let me take it back. Okay, so we have 2.5. Give me a minute. Okay, 2.5 here, and here we have 1.5 squared. When you use your calculator, you're going to get 22.5 centimeters cubed, and the answer is going to be here. So for this information here, we're going to use it in the next question, and I will return to this part so that I can explain further. So let's just move to the next page. Here they say a student investigated the feeding preferences of ground squirrels. They say design a laboratory investigation to find out which of the 3 types of nut ground squirrels prefer to eat. So since these are nuts, it's kind of food that they prefer to eat. It means we have to get some specific squirrels and expose them to the different types of nuts. And then we can calculate how many or count how many of these squirrels or how many nuts are going to be consumed by these squirrels. So I said we need to get various ground squirrels and allow them to access all the 3 types of nuts. The nuts, we have to make sure they are of the same size, and they should also be, so basically they have to be the same, and they have to be of various sizes. However, remember, I said same size, so in each category, if we should have maybe 3 centimeters from each of the nuts, and then 4 centimeters from each of the nuts, 5 centimeters from each of the nuts. If we can get those dimensions, so they have to be various sizes, but we have to make sure the sizes that are present in all groups are exactly the same. So length as well as width. And then this is in order to correctly record the type of nuts preferred and the most preferred sizes of the nuts. So remember, we are looking at which one they prefer, which length they prefer, maybe which width they prefer, so that we can make a fair conclusion. So lastly, we have to determine the order preference for the nuts by the squirrels. So whether squirrels prefer maybe the hazelnuts or they prefer the other nut or walnuts and so on. So we have to make a conclusion based on the observations we have seen in our experiment. Next year, they say, predict the results of this investigation giving reasons and use the information in the table to support your answer. I want to go back to the table. From the table, we can see that this one here has the highest energy content. However, it's really, really hard. It has a hard shell, so the squirrels have to input in more energy in order to crack this open because it has a hard shell. However, if they do, they're going to have a lot of energy. So it's better to be consumed when preparing for maybe winter seasons when they're going to require a lot of energy. While the acorns do not have a hard shell, so they're going to be easily accessible. It's easier for the squirrels to eat them. However, the energy content is really low, so that means they have to consume more of acorns than the walnuts. So these ones are better eaten when they're in plenty. These ones, if there is maybe scarcity or low food, then they have to eat this one more. So let's begin answering this. I say the squirrels would prefer to eat more hazelnuts because they have a small size, so they can easily be eaten by the squirrels themselves. Hazelnuts also do not have a hard shell or hard shell exterior, so they will easier eat them compared to the walnuts. So lastly, say since acorns store less energy, many would have to be consumed. This is what I told you about less energy content, so they have to eat more to gain the same energy requirement as they would if they had eaten maybe walnuts and so on. So this would be consumed to get enough energy required for hibernation. If they are to do this, they have to eat more of those nuts. Then I said walnuts, on the other hand, have a lot of energy to provide to the squirrels, so they would prefer to eat them before hibernation. This is when preparing for winter seasons when it's very cold. And I said since squirrels have sharp teeth, they can crack the walnut shell to access the high energy content. So in preparation for hibernation, they can use their sharp teeth to crack the nut and then be able to access the energy content or the high energy containing material inside the nut. Part C, they say suggest how ground squirrels evolved pouches. Now this is about evolution, so you expect there is going to be maybe a random mutation, variation is going to be created, there is going to be survival, there is going to be reproduction, and then passing on alleles to the offsprings, natural selection. So I said there was variation among ground squirrels. They had pouches of varying sizes. So remember, we have to include this. So maybe there was some mutation that occurred, creating various sizes of pouches. So as a result, squirrels with larger pouches managed to gather and store more food, while those with smaller sized pouches could not store enough food, maybe to survive during the winter. So the squirrels with the largest pouches survived because they stored enough food and they reproduced. They passed on the alleles they had, or the genes, for large pouches to their offsprings, leading to a higher allele frequency. Or you could add on here over generations. So the squirrels that had larger pouches were able to survive. So over generations, most of the squirrels evolved to have large pouches, or they evolved to have pouches. So this brings us to the end of question 6. Let's continue to question 7. So question 7 says, bacteria and fungi can both cause infections. The diagram shows part of a structure of a fungus. So here they've given us the cell wall, the cell membrane, the nuclei, ribosomes, glycogen granules, cytoplasm, mitochondria, and the Golgi apparatus. They say, how many of these organelles labeled in this diagram have membranes? So we can see there is, of course, everybody knows a mitochondria has a membrane. We know the nuclei or nucleus. Each nucleus is an organelle, as well as the Golgi apparatus. So there are only 3 among the rest of them. The answer should be a B. Next, they say explain why fungi are not classified as bacteria, plants, or animals. Everybody should know, because when you look back here, we see glycogen granules. So they can never be plants because plants do not store glycogen. And we can see here they have a cell wall. Animal cells can never have a cell wall, so they're not animals because they have a cell wall. And then because they have the mitochondria, they have the nuclei, they cannot be bacteria. So here I said they're not plants because of the presence of glycogen granules, and they're not animals because there is a cell wall. They're not bacteria because of the presence of membrane-bound organelles like mitochondria, Golgi apparatus, and the nuclei. So this could be very easy. Let's move on to the next part. Here they say, the graph shows the number of prescriptions of the antibiotic aminopenicillin issued in 1 year. They're going to say the graph also shows the percentage of E. coli bacteria that were resistant to aminopenicillin during the same year. So we see how these 2 graphs are changing. The first one is number of aminopenicillin prescriptions issued, and the next one is the percentage of E. coli resistant to aminopenicillin. So they continue down here to say, explain the relationship between the number of aminopenicillin prescriptions issued and the percentage of E. coli resistant to aminopenicillin during this 1 year period. So we can see as we do more prescription, the percentage slightly increases because, of course, that is kind of exposing them. Natural selection is going to occur. So I said, again, this is about explaining the relationship. So you have to begin by talking about the relationship. I say there is a correlation between the number of aminopenicillin prescriptions and the percentage of resistant E. coli. This part here is you talking about the relationship that explains that exists. And again, remember the question says, explain the relationship. You have to begin by acknowledging what kind of relationship they have. The relationship we see here is there is a correlation between the two. As one increases, the other increases. As one goes down, the other goes down. And then one increases, the other increases. So there is a correlation. And that is the statement you have to begin with before you do the explanation. So the next part is going to be explaining why the relationship is the way it is. So I said aminopenicillin acts as a selection pressure, killing bacteria that have no resistance, while those that are resistant survive and reproduce, allowing their offsprings to be resistant or to have resistance as well. So the resistant bacteria, then they will be increasing. So they will increase in number since they have less competition for nutrients after the death of the non-resistant bacteria. And then the percentage of resistant E. coli falls or after fall in prescription, because when the prescription goes down, of course, we expect them to be falling because aminopenicillin is no longer the selection pressure, but could be competition because there have been so many. When there is competition for resources because they have increased in number, of course, then they're going to begin to die. So here we say competition for other things like nutrients is, and then the non-resistant bacteria can increase in number too, leading to high competition for nutrients. In this part here, when we're using if you use the aminopenicillin here, maybe when it's increasing, we'll see that in this phase, the non-resistant bacteria are going to be dying while those resistant are going to be increasing slowly. And when you decrease the antibiotic, then we'll see that they're also going to be decreasing because the selection pressure is no longer the aminopenicillin. So it allows the non-resistant bacteria to increase as well. So competition causes the decrease in the number of the resistant bacteria until the increase in antibiotics occurs. The non-resistant bacteria will die, and then that allows for the resistant bacteria to be able to increase in number. So let's continue to the next part. Part C, the results of a survey are shown in the diagram. So here they say 90% of doctors have felt under pressure from a patient to prescribe antibiotics, and 70% of doctors prescribe antibiotics when they are unsure if they are treating a viral or a bacterial infection. 49% of doctors prescribe antibiotics once a week or more without knowing whether they are medically necessary. And 44% of doctors prescribed antibiotics to get a patient to leave the surgery. So we can see all this information. And then they ask, explain why the results of this survey are a cause for concern. Of course, this is about resistance, antibiotic-resistant bacteria. So let's see. I said, if doctors ignore the code of practice, remember we are referring to the code of practice that they should prescribe antibiotics only when necessary. So if they ignore the code of practice in prescribing antibiotics, this will cause concern because the number of antibiotic-resistant bacteria will increase. Antibiotics act as selection pressure, killing bacteria without resistance and enabling those with resistance to increase in number. So as a result, the resistant bacteria increase in number, and the available antibiotics will no longer work on the resistant bacteria strains, leading to a lot of people dying from exposure to bacterial infections. So the code of practice that the doctors have to adhere to is that you only prescribe antibiotics when necessary, and actually to the patients as well. If you're given antibiotics, you have to make sure you complete the dose without a fail, because if you do not, then you give a chance to the bacteria to be exposed to half a dose, not completely destroyed, but there can be mutations that can occur on less dosage, exposure to less dosage, and that can lead to antibiotic resistance as well. So it is required that doctors do not prescribe a lot of antibiotics, and if they do, then it could lead to antibiotic resistance. So this brings us to the end of question 7. Let us go to question 8. Question 8 says, humans can have one of 4 different types of blood types. There is A, there is B, AB, or O. So they say blood type is determined by antigens present on the membranes of red blood cells. The table shows which antigens are present in each blood type. So we can see blood type A has antigen A only. They've given you the diagram here. And type B has antigen B only. We can see that. AB has both antigen A and B, and you can see the A's are here and the B's are there. And then O, neither antigen A or antigen B. So they're going to say, blood transfusions are used in the treatment of some diseases. A blood transfusion involves taking blood from a healthy person and putting it into a person needing the treatment. Which blood type can be used for all blood transfusions? Of course, it has to be blood type O because it doesn't have any antigens and it's not going to cause any immune response in the body. So let's continue. Explain what will happen if the person with blood type A is given a transfusion of blood type AB. So of course, the person who has blood type AB has both A and B antigens, while this one just has A antigens. So the B antigens will be detected as foreign antigens. So I said, when a person of blood type A receives blood from a type AB person, the B antigens present on the membranes of the red blood cells in the transfused blood will be recognized as foreign antigens. This will trigger the immune response, leading to the release of antibodies by lymphocytes in the blood. Of course, you guys know lymphocytes work, but when the lymphocytes release antibodies, these antibodies work by causing opsonization, agglutination, which later is cleared out by phagocytosis. So I say this will cause the destruction through opsonization and agglutination of the red blood cells with the foreign antigen, and the memory cells can be created to produce a faster response if blood with similar antigens is given in the future. So if a person is exposed to AB antigens, both of them, yet their blood is A, the immune system is going to create or lead to formation of memory cells that will react faster in the future if they're exposed to the same foreign antigens. So here we see the next part says, bacteria live in our intestine, which we call the gut flora. The membranes of cells lining our intestines have molecules similar to antigens on red blood cells. They say, explain why bacteria living in our intestines are important. They are important because, of course, this is a common question. They will outcompete or they compete with the pathogens for space and nutrients, and most of the time, they do outcompete the pathogens, protecting us from infection or from disease. So I say that gut flora compete with pathogenic bacteria inside the gut for nutrients and space. You should write this specifically. It's very important. This is what they compete for. And then this reduces the growth of the pathogens, preventing infection by the pathogenic bacteria. Let's continue to the next part. They say, both the molecules on the membranes of the cells lining our intestine and the antigens on red blood cells have sugars attached to protein molecules. So they say, bacteria living in our intestine secrete an enzyme that separates the sugars from the protein molecules. Suggest why bacteria living in our intestine secrete this enzyme. So they do so that they can absorb the sugar into their cells to use it for respiration in order to obtain ATP. This is really basic. You remember any organism, basic or any cell, it has to be able to carry out respiration. More so for those living in the human body, there should be respiration because that is the way they obtain energy for cellular processes to be carried out. So they will need respiration. So they do that. Break off. Remember here they've said these are protein molecules that have sugars around them. So they're going to break off the sugars, leaving the protein molecules alone, and these sugars are going to be used for respiration. Finally, here they say, explain why this enzyme may be useful in blood transfusion. So I say this enzyme can help to remove the antigen on the red blood cells. So if this is removed, then of course the red blood cells are not going to be detected as having foreign antigens. So any blood type can be used for transfusion without triggering immune response. This brings us to the end of question 8. I will continue to question 9. Question 9. The photograph shows the moose. Here, this is the moose we're looking at. So they say, moose are large mammals that eat grass and the shoots of plants. The number of moose is declining in parts of America. The graph shows the number of moose in one part of America from 1994 to 2016. We have on the vertical axis the number of moose times 10 power 3, as we can see. And on the x-axis, we have the years. For my estimate, it was about 7.6 times 10 power 3 to about 3.4 times 10 power 3. The first question states, in 2016, the moths were not counted. The value shown on the graph is an estimate. Describe how the data were used to estimate the number of moths in 2016. So here I could say they could have used extrapolation. Using the previous year's data, you could extrapolate to estimate the data for 2016. You could use mean yearly decrease as well as that. And then you could draw a line of best fit and also be able to get that. So this was a 1 mark question. Any of this would be okay. Let's continue. They say calculate the percentage decrease in the number of moths from 1994 to 2016. Now, you remember I had 7.6 times 10 power 3 to 3.4 times 10 power 3. So I'm going to use this data and then divide by, remember they're asking us for percentage decrease. So it's going to be the change that has occurred, which is a decrease, divided by the original times 100. So it gives us about 55.3% if you round off to 3 significant figures. And that is going to be the answer here. The next part says, it has been suggested that global warming is responsible for the decrease in the number of moths from 1994 to 2016. So here they say, sketch a graph to show this relationship. Remember, they have said global warming is responsible for the decrease. So this is going to be the dependent variable. And then global warming is going to be the independent variable. Because, of course, global warming is going to be increase in temperature. So remember, this is causing that. So the number of moths should be on the y-axis because you're going to be counting them. And then the temperature, which is increase in heat or temperature, should be on the x-axis. And then since we have a decrease, I just did like that. That is a sketch. It doesn't have to be perfect. So let's continue to the next question. They say, ticks are parasites that live in the fur of moths. Ticks are thought to be responsible for the decline in the number of moths. The table shows the frequency distribution of ticks in a population of moths. So we can see, I'll just continue down here. And then they say, calculate the percentage of moths that have 50,000 or more ticks in their bodies. So we can see 50,000 or more, they're going to be here. But remember, we're counting a percentage. So it has to be, add everything up here, you get 41. So it's going to be 41 divided by the total. All these, when you add them up, you get 214. So 41 divided by 214 times 100 gives us about 19.2%, which is the percentage of the moths that have more than 50,000 or more ticks. So let's continue. Here they say, the diagram shows the life cycle of a tick. So you can see, of course, if we are to begin with this one in June, the female is going to, so after several weeks of dormancy, the female lay their eggs, starting a new cycle. So the cycle begins that summer. Winter tick larvae hatch from clusters of eggs that have been laid on the ground. And then we go on. Here in the autumn, the larvae climb into the vegetation, latching onto any moth that passes by. Once attached, they feed for the first time and molt into a nymph. The larvae cannot do this if they are covered in snow, so they have to do this in the autumn. And then in the winter, after a few months of dormancy on their host, the nymph feed again and molt into adult nymphs. And then the feeding begins. This is like winter feeding, again as the tick mating season gets underway. And then finally, in the early spring, the female tick fully engorged on blood drop off the moth. The ticks die if they fall onto the snow. So this is the cycle we have seen. I'm going to go down here to see how we can answer some of the questions. So they say, the ticks feed on the blood of moths. One tick can remove 200 to 600 times its body mass. And the tick irritate the moth. The moth will scratch against trees, causing large clumps of fur to fall off. Remember, this is winter. The fur is very important to the moth. So if it falls off, then they're going to be affected. They say, explain how global warming might affect the life cycle of ticks and result in the decline in the number of moths. They want you to use all the information in the question to support your answer. This is a 6-mark question, so we have to go all the way. I said, global warming leads to increase in the temperature of the Earth's surface, and this can result in warmer or shorter winters. So if there is global warming, we could have warmer winters, and the winters could be shorter. So the warmer conditions will lead, let me just write a lead. Sometimes use the iPad, some of the things that are deleted slowly. So I say, Warmer conditions will lead to the decrease in the in the time taken for the ticks to morph. Morph is like change into adults and mating. So we go on to see more nymphs will morph or change into adults, and the females will lay more eggs. That is more. And the availability of less snow due to warmer conditions lead to fewer death of the ticks. Remember, they say the ticks fall off. If they fall on the snow, they will die. So here they would be falling on less snow or no snow at all. So there is a higher chance of survival. And as a result, more ticks will inhabit the fur of the mouse, requiring more blood. Remember, during the cold conditions, the mouse are going to be inside the fur. So if there was a lot of reproduction going on and a lot of ticks are going to be produced, or they will latch onto the fur, and then they will be able to drink more blood from a single mouse, leading to greater blood loss and therefore a higher chance of the mouse dying. So the mouse will lose a lot of blood and nutrients required for respiration, and some mouse can die due to lack of nutrients or anemia, or due to lack of energy for hunting. So here, if there is excessive scratching onto surfaces by the mouse due to tick irritation, this can lead to excessive loss of fur to keep them warm during the cold days. And scratching also leads to open wounds, which of course lead to pathway for pathogens to infect the mouse. So there are so many things that can lead. Either the mouse could die due to coldness, it could die due to infection, or it could die due to excessive loss of blood. So all these things can lead to the decrease in the number of mouse within the specific area. This brings us to the end of this paper. Thank you for watching with us, and please do not forget to subscribe. As I said previously, I will put the link to the first video in the description box. Thank you. Bye-bye.